Hi everyone, Brenda Gustin here, and this week we are going to be talking about the control of microorganisms. And so we've talked about how they grow, and now we're going to talk about how we stop them from growing. And this is something you have some basic knowledge about. The chapter has lots of examples of things that you'll be familiar with, and I'll run through those. And there's definitely some that maybe you're not quite as familiar with. So the chapter starts off being very heavy in vocabulary. So I highly recommend you practice your vocabulary lists. I know a lot of the adaptive learning questions, if you just know the definition of the word, you'll be able to answer the question. In fact, here's an NCLEX question that's quite good, but maybe you don't have the information yet to answer it. So come back to that one. So we're going to talk about, I'm in section 9.1, and it just asks you to define a lot of um, the vocabulary. And it says the methods of control used outside the body are designed to result in four possible outcomes: sterilization, disinfection, decontamination, also called sanitization, and antisepsis, also called degermation. And so we're going to talk about those. In fact, they put these vocabulary words in a table, and there's some really nice pictures here that you can look at. So I highly recommend going back and looking at those. They give you some really nice examples. In this table, it says the ones that have a pink background are capable of sterilizing. And so sterilization is going to be our first vocabulary word, that it says it's a process that destroys or removes all viable microorganisms, including viruses. So it inactivates viruses. Essentially what it does is it prevents all microorganisms from being able to reproduce. It kills them. And viruses, it inactivates them because they're not actually alive. And so it says the word sterile should only be used in the strictest sense. You can't be kind of sterile. You either have to be sterile or you're not. You're free of microorganisms or you're not. It's kind of like being kind of pregnant. You got to be one or the other. There's no halfway. Generally reserved for inanimate objects, because trying to sterilize living tissue would most certainly damage the living tissue itself. It would kill the living tissue. So we don't really use that word for living tissue. And it gives you some examples here of how we sterilize things. And we're going to be talking about that as we go through the topics. Disinfection. And, I like to put disinfection and antisepsis together. Disinfection, a physical process or a chemical agent to destroy vegetative pathogens, but not endospores. And so remember that some bacteria made endospores, and they are highly resistant to environmental conditions, to chemicals and physical control. And so when we're talking about disinfection, and we do a lot of disinfection around our houses, we're targeting using being able to kill the vegetative pathogens, the bacteria in the form that they're actually growing. We're not trying to kill endospores. We're not sterilizing by disinfection. And so the other thing about disinfection is typically we're talking about inanimate objects. And the reason for that is the methods for disinfection tend to be would be potentially harmful for living tissue. So you don't typically disinfect your skin with bleach. Sometimes we use iodine, but or heat. These would be too vigorous for skin. And so we save the word antisepsis for reducing the number of vegetative pathogens on human skin. So again, it's not sterilization. We're not getting rid of endospores, but we're just reducing the number of pathogens or microbes in general on human skin or other living tissue. And so antisepsis can be scrubbing or using some kind of chemical. So alcohol is a good antiseptic that we've all used for hand sanitizers, surgical hand scrubs, but even washing can contribute to antisepsis. Now there's another term, here we go, kind of in between. We use this word decontamination, or you might hear it called sanitization. I use the word sanitization more often. And we hear about sanitizing things. And what that means is it's talking about inanimate objects, and typically this is something we would do in it says restaurant, dairy, like in a commercial setting, that you're going to reduce the number of microorganisms. You're not going to sterilize, but it's like cleaning the dishes. You're going to sanitize them, and so it's going to remove microorganisms as well as debris. And so that would be things like soaps, detergents, dishwashers, things like that. And so there are protocols in commercial settings for sanitizing, for keeping the restaurant clean, for keeping the hospital surfaces clean. And so we might decontaminate, or in a more vigorous way, if you're really trying to reduce the number of microbes, in a more vigorous way, it would be disinfection on inanimate objects. And so here's part of where we're heading. We're going to actually talk about the physical and the chemical ways that we control the growth of microorganisms. Now they put mechanical here, but I kind of would have put mechanical with physical, but it doesn't really matter. But they're showing you the different ways to apply these terms. So here we can actually use heat and to sterilize, to eliminate all microorganisms, whether it's dry heat or moist heat. We can use radiation to sterilize. When you look at the chemicals, we can there's some gases that we can sterilize with. There's actually a liquid or two that can sterilize, but most of the others do disinfection or antisepsis. So we're also going to talk about filtration. And so this is we're going to expand on this with some specific examples. The second outcome I have is here's identify the microorganisms that are most resistant and those least resistant to control measures. And so this table, this diagram is a really good reference for that. When we're talking about microbial control, you want to choose your microbial control agent to match your pathogen. What are you trying to accomplish? If you're trying to sterilize something, eliminate all microorganisms, in the case that you would do for utensils for a surgical procedure, or containers when you're going to grow bacteria in a laboratory, your most resistant structures that you have to think about are endospores. Again, remember we said only a couple kinds of bacteria make endospores, but these are common bacteria. So the criteria for sterilizing something is, are you able to inactivate endospores? Now you'll notice on here it says prions are actually more resistant to microbial control than endospores. But prions are not something that are normally in our environment that we have to be concerned about. The only way they actually grow is if they get into brain tissue. So even if we got some prions in our flask where we were growing bacteria, they wouldn't have any impact on it. So, and again, the likelihood they would be there would be extremely small. It's not very likely at all. So the standard for sterility is bacterial endospores. Prions actually take above boiling, above conditions that are more rigorous than those to kill endospores. So we're not really going to talk about that very much because that's very unlikely to be an issue in your experiences. So if we can inactivate endospores, we've been able to sterilize. Notice here the next hardest thing to kill by microbial control are bacteria in the genus Mycobacterium. And we've mentioned these bacteria before. These are the bacteria that cause tuberculosis and leprosy. And they happen to have this waxy wall. You might remember in lab we did acid fast stains, and we were looking for this waxy wall. Well, that makes these bacteria very resistant to drying out in the environment and resistant to control. So they're the most difficult bacteria to inactivate next to those that have endospores. Here's another one that you've heard of, Staphylococcus, and maybe one that you should be familiar with, Pseudomonas. Both of these are common pathogens. They're in our environment, and they tend to be somewhat more resistant to microbial control than most gram-negative and gram-positive bacteria. Remember, we said some protozoans make cysts, and the cysts help them survive environmental conditions. So they're one of the structures that is a little bit more resistant to microbial control. And then we start getting down into Some of the other things we've heard about gram-negative bacteria, gram-positive, um, fungi and their spores, viruses. And you might remember we said enveloped viruses are more sensitive to microbial control than non-enveloped. So the take-home messages out of here are endospores. The ability to inactivate endospores is really our definition for being able to sterilize. Mycobacterium in particular are highly resistant to microbial control also. Here's a little background and note about prions, and you can read that. I'm not going to go into it, but you might find that interesting. Here's just a table comparing what's required to destroy endospores versus vegetative cells. Remember, vegetative cells are the actively growing cells. So it says moist heat at 120 degrees Celsius, and this is for about 20 minutes, are able to kill endospores. And remember, 100 degrees is boiling. To kill an actively growing cell that's not making endospores, it's about 80 degrees Celsius. Higher doses of radiation are needed to inactivate endospores. Higher doses of certain gases that we use to sterilize and certain liquids we can use to sterilize are needed to inactivate endospores. So endospores are much more resistant to environmental pressures than vegetative cells. Okay. Number 3 on your learning outcomes says compare the action of microbial control agents that are cidal versus static. And the terms I have there, the root of the words cide means having the capacity to kill, and static means to stop growth. Here we go. Stasis or static means to stop growth or to stand still. And so you can use those roots of those words to create a lot of words. And if you look on your handout, you see I have bactericidal, any treatment that's able to kill bacteria. And it doesn't mean it necessarily sterilizes, but it's able to kill bacteria. It's kind of a general term. Bacteriostatic, a treatment that will stop the growth of bacteria. A bactericide is a chemical or a treatment that is bactericidal. So you can see how those words can be used. A fungicide, a treatment that kills fungi. A virucide, a treatment that kills viruses. So if you can get those roots in your mind, cide and static or stasis, you can create a lot of words out of those. There's even, here it says germicide and microbicide are actually just real general terms for the ability to kill any germ or microorganism. So anyway, a couple other words we have on there. It says sepsis is the growth of microorganisms in the blood or other tissues. And you probably know if you put an A in front of a word, it makes it not. So asepsis is going to be something that prevents the entry of microorganisms into the body. So you're going to use, in the laboratory, we use aseptic techniques, not dirty, techniques that prevent the introduction of pathogens. Let's see, I think we got all of our words there. Oh, we have one more word, antisepsis. We had that with antiseptic, so we're good with that. All right. All right. All this stuff is good stuff. It's all interesting. It's just we don't have time to do it all. So I think I'm going to stop. Actually, I'm not going to stop. There's one more. It says distinguish between the physical and chemical control agents and name 4 categories of cellular targets for physical and chemical control. So when we're talking about the rest of the lecture, we're going to talk about physical ways to control microorganisms. That's manipulating the environment, and chemical ways, using certain chemicals, whether they're liquids or gases. And so we're going to group everything that we're doing into one of those 2 categories. This just shows you what are some of the targets. And so if we can target different things in the cell, I mean, what do cells need to live? So if you can inactivate those cellular functions or structures, you're going to kill the cell. It's going to cause a cell death. So remember, in bacteria, they need a cell wall. So if you inactivate the cell wall, you will be able to kill the bacteria. Cytoplasmic membranes, remember that controls what goes in and out of the cells. So again, if you inactivate or if you disrupt the cytoplasmic membrane, you can kill the cell. Cellular synthesis processes. What are they doing for their metabolism? Metabolic pathways, making DNA, RNA, and proteins. All of that, any metabolic processes are, if you can disrupt them, you can kill the cell. And finally, remember, we talked a lot about how do you denature proteins. And so if proteins are denatured, your enzymes no longer function or your receptors no longer work, things like that. So any of these can be targeted by the chemical and physical control methods that we're talking about. That is the end.